Now let's discuss exercise on chapter 1, which is genetic inheritance and genetic control, part B, structured question. Question 1. As you can see from this diagram, it shows you the crossing between a pure breeding line of red snapdragon to a pure breeding line of white snapdragon. So let's say, for example, for the pure breeding red snapdragon, we give the genotype, or we assign the genotype for the red as CR. Okay, and as for the white snapdragon, we assign the genotype as CW. So as you can see from the genotype, uh, CR does not mean that it has 4 alleles. Okay, CR is 1 allele. Okay, and CW is 1 allele. Okay, so basically C stands for the genes that code for flower color. Okay, so gene that codes for red flower color, genes that code for white flower color. Okay, so for the red plant, that is the parent for that has red flower. Okay, so the gamete will be CR. As for white, the gamete will be CW. So when the gametes fertilizes each other, so it produces F1, okay, generation that has a new phenotype that is different from both parents. Okay, so the phenotype of F1 generation is all pink. Okay, so so basically the genotype of the F1 generation is heterozygous, CRCW, and the pink phenotype of F1 is actually the phenotype that is in the intermediate phenotype from the two parents. Okay, so the F1 generation, then you allows the F1 generation to undergo self-fertilization to get the F2 generation. So for the F2 generation, 25%, okay, 25% of the population of F2, or one quarter of F2 has the red flower phenotype. 50%, okay, or one half of the population has the pink flower phenotype. Okay, so they are heterozygous, and another one quarter, or 25% of the F2 generation has the white flower phenotype. Okay, so basically when you produce a phenotype in the offspring generation that has intermediate phenotype from the initial parents. Okay, so this type of inheritance pattern, we call it as incomplete dominance. For the next question, you have to give reason for your answer in A1, which was the answer for A1 was incomplete dominance. So why is the inheritance is incomplete dominance? It is because the F1 generations, which are heterozygous, okay, they show intermediate phenotype, okay, or phenotype that are intermediate between the phenotype of the homozygous parent. So that is for 1 mark. So the keyword here is intermediate phenotype, or phenotype that are intermediates between the phenotype of the purebred parents. Or another way you could explain for your answer for the same 1 mark is that the F1 generation shows the blended color, okay, which is pink, which is the intermediate color between red and white of the parental generation. Or another point that you could write, okay, for the same 1 mark is neither allele, which is neither of the CR or CW allele, is dominant over one another. Okay, or both alleles are partially expressed. Okay, so CR allele and CW alleles are partially expressed, so that it produces in the F1 generation pink flowers. For question 3, you have to determine the genotypic ratio and also the phenotypic ratio of the F2 generation. So basically for every crossing, you have to know how to determine the genotypic ratio and also phenotypic ratio. And as you can see from the diagram here, I already wrote, okay, the genotype for every generation from the parental generation, F1 generation and F2 generation. So basically for the genotypic ratio, as there is 1, okay, CR, 2 CR/CW, to 1 CW/CW, okay. And as for the phenotypic ratio, it produces 1 red, okay, to 2 pink, to 1 white. So for incomplete dominance, basically the genotypic ratio and phenotypic ratio, they are the same, which is 1 to 2 to 1. Make sure in for example in the exam, if it asks you to write the genotypic ratio and also the phenotypic ratio, do not just write 1 to 2 to 1. You have to write clearly: 1, what is the genotype? 2, what is the genotype? 1, what is the genotype? You have to write the genotype. Same goes to the phenotype. You have to clearly write the phenotype for each ratio that you wrote. Okay, do not just simply write 1 to 2 to 1. That will not give you mark. Okay. Question B, it says Haras has AB blood type and his sister has O blood type. So what are the genotypes of the sibling parents? So you have to determine the genotype of the parents. So to make it easier, you just draw the crossing of the parental generation and the offspring generation, the F1 generation, which produces Haras and his sister. So Haras here, he has AB blood type. Okay, so it means that the genotype of Haras is IA/IB, and as for his sister, which has blood type O, so the genotype of his sister is IO/IO, or you could write it as I. Okay, so it means that for example here, you could write this is the father, okay, so this is the mother, or you could do it as vice versa, whichever you can. Okay, so obviously, in order to get the genotype IA/IB, it has to occur between the fertilization of sperm and eggs that carries different alleles. Okay, IA sperm that it may be sperm that carries IA allele. Okay, sperms that carry IA allele that fertilizes the ovum that has the IB allele. Okay, so that is one of the possibility. So the fertilization between sperm that carries IA allele and eggs that carries IB allele produces the genotype IA/IB that produces Haras. Okay. The next possibility is that the fertilization of sperm, okay, that carries the IO allele, okay, IO allele, that fertilizes the eggs that carries the IO allele. So this produces his sister that has blood type O. Okay, so when you have determined the gametes for both of the parents, now you can determine or know the genotype of the parents. So therefore, the genotype of the male parent or the father is IA, IO, and the genotype of the mother is IB, IO. Okay, or vice versa. So it is the same. So for the 2 marks, okay, so if you write correctly, the genotype of one of the parent is IA/IO, so you get 1 mark. And if you write correctly the genotype of the other parent as IB/IO, you get another 1 mark. Question C. In one variety of maize crop, a gene at one locus can occupy the dominant K allele, okay, that inhibit kernel color, while the recessive small k allele permits kernel color when homozygous. At a second locus or a different locus, the dominant R allele expresses the red kernel, while the homozygous recessive genotype small r expresses the purple kernel. So from the statement given in the question, there are basically two genes. Okay, the first gene, it controls basically the color of the kernel. Okay, so the first gene, it controls kernel, the expression of kernel color. So there are two alleles involved, which is the dominant K allele and the recessive K allele. So for the dominant K allele, it inhibits kernel color. Okay, so the individual that has the homozygous dominant phenotype and also heterozygous will not permit kernel color. It inhibits the expression of kernel color. And as for the recessive allele, okay, that permits coat color, individual with a homozygous recessive genotype, okay, will allow the expression of kernel color. And as for the second gene, it controls kernel color. Okay, so there are two possible alleles, which is the dominant R allele that produces red kernel. So individual that has homozygous dominant and also heterozygous produces red kernel. And as for the recessive allele, individuals that are homozygous recessive will produce purple kernel. Okay, so basically when in the question it gives you so many information, you have to extract out all the information so that it is easier for you to understand the question. So let us just test, okay, our understanding. So if the genotype is capital K, capital R, which is both genes, they are in the homozygous dominant condition. So what is the phenotype of the offspring? So this produces offspring that has what? Colorless, that are colorless basically. So although the second gene it is dominant, homozygous dominant, but because the first gene is homozygous dominant, it will inhibit the expression of red color. So the kernel color will be colorless. Okay, so same goes to this phenotype. Okay, sorry, genotype. So if the genotype is as long as it has the dominant K allele, okay, so this produces colorless kernel. Same goes to this genotype. Okay, so because it has the dominant K allele, so this also produces colorless kernel. And as for this genotype, as you can see, because the first gene is in the homozygous recessive condition, so this produces red kernel. So same goes to this one, because the first gene is homozygous recessive, so this produces purple kernel. So basically the pattern of inheritance shown here is epistasis. There are two genes, okay, one gene will control the expression of another gene. So for question 1, complete the Punnett square below that shows the crossing between two plants that are heterozygous for both loci. So this is worth 3 marks. So for the first mark that you get is when you write the genotype of both parents correctly. So in the question it says that both parents are heterozygous for both loci. So therefore the genotype of both parents are capital K, small k, capital R, small r. Okay, so same goes to the second parent, capital K, small k, capital R, small r. So this is 1 mark. Okay, so you write the genotype of both parents correctly is 1 mark. For the second mark, if you write the gametes, okay, all the gametes for both parents correctly, so you will get 1 mark. Okay, so the gametes for both parents produced are the same because they are heterozygous for both loci. So the gametes are capital K, capital R, capital K, small k, capital R, small k, small r. Okay, so same goes to the male gametes. So that is for 1 mark. For the third mark, if you write correctly all the genotype of F1 generation correctly, okay, so all the genotype within the Punnett square you write correctly without any mistake, okay, the combination of the genotype are correct, so then you get another mark. Okay, so that is the answer for question 1. Okay, so for question 2, you have to determine the phenotypic ratio of the F1 offspring. So from the Punnett square, you can easily count the phenotype that produces in the F1 generation. Just remember, every time when you have determined the each of the individual genotype, just straight away write the phenotype just below the genotype. Okay. So it is easier for you later to count all the possible phenotype that are present in the offspring. So basically, the answer that you will get for the phenotypic ratio is 12 colorless, okay, to 3 red to 1 purple. Okay, so this is the phenotypic ratio that you will get for this question. So just remember, for epistasis, you could get different phenotypic ratio based on the condition given in the questions. Okay, so you have to read carefully what are the condition given in the questions. So the answer can be different from this ratio. Okay. So satu lagi tips yang saya nak bagi tahu kat sini, bila awak buat Punnett square yang ada empat different gametes kat sini dan you produce 16 different combinations of genotype, dia bukan bermaksud yang ini adalah F2 generation. F2 tu adalah daripada Mendel's Dihybrid cross. Okay, so Mendel Dihybrid cross dalam Punnett square tu yang awak produce adalah F2 generation. So in this case, awak kena faham konsep yang mana the parental generation is already in the heterozygous condition, and in the Punnett square is the F1 generation, not the F2. Okay,